Hi, for this experiment, we're going to show you how to break proteins. But first, what is proteins? Proteins are required for the proper functioning of all living things or living organism. Uh, Proteins are large compound molecules that are made up of many amino acid chains. Proteins fold in a very specific ways. They are the way they fold control what the proteins are able to do. Proteins aid in a movement of other molecules, responds to the signal and make reaction happen more quickly and replicate the DNA, among other functions. But if proteins lose their specific fold shape, they are not able to do their, their work properly. To maintain their forms, proteins required particular or specific condition. Most proteins in our bodies rely on us to keep warm or healthy body temperature, uh, stay hydrated, and consume enough of specific nutrients like salt. Some of our proteins may not function or not at all if our bodies are unable to maintain certain conditions. Most organisms create molecular chaperones that help other proteins, and molecules continue to work even when the conditions are becoming difficult to tolerate. Uh, the function of a protein is undone when it is subjected to conditions that are too far outside of its tolerance range. Denaturing or basically breaking a protein is deterred in this process. We denature proteins all the time when we cook food like eggs. In this activity, we will use egg or process to denature egg proteins in two ways: by cooking them or and by exposing them to concentrated alcohol or ethyl alcohol. They believe that the appearance of egg will be the same or maybe vary depending on how the protein contains or hold are denatured. So let's find out in this activity. For this activity, the materials that we need, first is the electric kettle to boiled water. Next is pork, scissor, for same size container, a glass container, same size, uh, egg white, ethyl alcohol, tap water, room temperature, and last is a measuring cup. Uh, First, we're going to uh, pour ethyl alcohol, uh, 75 ml. to this container for letter C. Next is tap water. I already washed this when I use alcohol. Same size, 75 ml. Next is we're going to cut the egg white that we will add to hot water as soon as possible. But first, we're going to cut before we put hot water in this container. Next is hot water. Same size. And then next is we're going to put in the hot water alcohol, in tap water, and air control. We will leave this for 2 minutes and then we will mix it using a fork. After 2 minutes, we're going to mix using fork. First, the hot water. Then we will wash this. Next is alcohol. Next is tap water. And last is our control. We will leave this for 30 minutes. We will observe the result. After 30 minutes, the egg white start out clear. They are almost 90% water, but the 10% is packed with protein. Egg whites contains more than 50% of protein found in the egg. The main protein in egg white is called albumin. The small folded proteins in the egg white don't take up much space, and the gel-like egg white looks clear. The control egg show us that when we left room temperature, the egg whites stay clear, meaning the protein maintained their original shape. These proteins were not denatured. Next is hot water. Whenever eggs are cooked with heat, the egg white turn from the clear to white, and the gel becomes more rubbery as the heat denatured the protein in the egg white. It broke apart some of the bond, mostly hydrogen bonds, that were holding the protein in the original shape. The proteins unfolded, taking up more space, turning the gel white and hardening them in the place next to one another. Next is alcohol. Alcohol also denatured pure proteins. It does this the same way as the heat, by breaking the bonds that hold parts of the protein in the folded shape. Sometimes the alcohol molecules bond directly to some of the parts of the protein, disrupting the normal way of the protein would bond to itself. The alcohol is called the bond disruptor. The proteins again unfolded, taking up more space and hardening in the place next to one another. This process took much longer with alcohol than we did to the heat. However, the longer time for denaturation with alcohol is simply because it spread more slowly than heat. The alcohol had to diffuse, um, move through the fluid into the egg in order to affect the protein it touched. And lastly, the tap water or the room temperature water. Sometimes in this experiment, room temperature water or tap water has a small denaturing effect on some of the egg white. It acts in the same way by breaking bonds, but its effect isn't nearly as strong as alcohol or the hot water. Uh, that's all. Thank you.